resume the recording here, and we'll get started. Okay, there is a chat box in the Zoom meeting space. If you kind of mouse over the bottom section of your browser or your computer, a lot of times you'll see this menu sort of pop up, and there's an option to send a question to me. So during the course of the webinar, your mics are muted, but if you send something through the chat, I can I can see it and then try to answer it. What I'll try to do during this session is I'll try to see those chats as they pop up, but most of the time what ends up happening is I kind of go through the webinar, and then at the end I'll address the questions. Okay. So my name is Jack Panopoulos. I'm an application scientist with FlowJo. I've been there for about 5 years now, and seen a lot of different data sets, which is really pretty cool. FlowJo is now a subsidiary of BD. Everything's going to continue like it has been, so don't worry about that. We still give these webinars, and we're still going to train you how to use FlowJo, and FlowJo will continue to be rolling along like it always has been. Anyway, as far as getting help with FlowJo, aside from today's session where we'll do a kind of a walkthrough through a phenotyping experiment, I'll show you how to set up some layouts and get some data out of FlowJo, how to create tables, and how to generate a compensation matrix, among other things. There is additional help if you simply email techsupport@flowjo.com. So anything that you're looking for in the program, something you can't find, maybe you run into some sort of a snag in the program, by all means, just send an email to tech support. They're in the office from 7 a.m. till about 5 p.m. Monday through Friday, and they are located in Ashland, Oregon, on the west coast of the U.S. Okay, we also have additional materials online. If you just go to flowjo.com, we have a tab on our website that's labeled Learn. Underneath the Learn tab, you will see lots of different sort of sub hyperlinks that will take you to different sections. So we have video tutorials, we have online manuals, we also have these webinars have been recorded. There are some webinars that we've done that are very specific. So if you're looking to figure out how to do t-SNE analysis or use some of the new clustering tools that we have available, we do have webinars that cover those topics specifically on our website, on our recorded webinars page. And all of that stuff can be found just by going to flowjo.com and clicking on the Learn tab. We do have some new tools. So any of you out there that might be thinking about doing single-cell RNA sequencing, we do have an application, a new application called SeqGeek, which is available. The nice thing about it is that you don't have to use all, which is super cool, especially if you're not very familiar with programming languages. So that, I think, is a huge boost to the field. You can also review or you can also analyze bulk RNA experiments using SeqGeek as well. So it's not really restricted to single cell. It's just, that's what we started with. Anyway, we also have another product called FlowJo Envoy, which is a web-based application for basically project management. It's nice because it presents you with a dashboard of your progress on a given experiment. And it keeps all of the data and all of the communication about the data in one place, which is really nice. Pardon me. Which is really nice because when the PI asks you to go, find so-and-so's data who left the lab a year ago, it's been very difficult to do that when you're just looking kind of at a regular server. But if you use FlowJo Envoy, it makes that search a lot easier. We also have additional plugins and other tools that I'll kind of introduce. I won't really go through them all, but I'll show you where they are and maybe a little discussion about what they do. A lot of the new plugins, as we release them, are on the FlowJo Exchange. So again, you can access the FlowJo Exchange by going to flowjo.com if you want to use one of the sets of newer tools. Okay, so easiest way again to find help, just simply send an email to tech support. They'll be more than happy to answer your question. Yeah, I'm going to end the slideshow there, and we'll jump right into FlowJo here. And what I'm going to do in this case is I am going to bring in the data files that I have on my system. So I have a series of folders here. One of them I've labeled the immunophenotyping experiment. If I double click on the folder, inside the folder is another folder, but there are a bunch of FCS files. Okay? FCS files stands for flow cytometry standard. These are the types of files that FlowJo reads. FlowJo can also read LMD files, which are produced by a lot of Beckman machines produce LMD files. And we can also read MQD files, which are generated by MaxQuant. But most cytometers, regardless of who the manufacturer is, will produce FCS files. And if you had the choice of the file type export, I would always go with FCS. And if you have the option to do the latest FCS version, like FCS 3.1, right, or FCS 3, by all means, go ahead and export it in that manner. Okay, when we bring it into FlowJo, it's up to you. You can bring in the individual FCS files, or you can simply just drag in the folder that contains the FCS files, and FlowJo will immediately populate them here to the lower section of your workspace. Okay, so this is the FlowJo, what we refer to as the FlowJo workspace, the entire application essentially in this pane. At the very top of the workspace are a series of tools. So you have undo and redo buttons here, right? And then you have a little tube with the plus. This is to add samples. So if you would rather navigate your file system rather than using the Finder or the Explorer. Okay, I should mention that FlowJo version 10 is compatible with both Mac and PC, so it doesn't matter which system you use. You can analyze the data on either PCs or Macs. Okay, and the functionality is very much the same. There's very few differences between the two. Okay, if you want to navigate your file system, you just click on the tubes with the plus, and this will actually open up like a Finder window, very much like the one that I had just opened, and you can go ahead and find the files that way. Okay, there are some other tools up here that we're going to make use of today, especially creating groups. The comp wizard, how to generate a compensation matrix. And then we have something called the table editor and layout editor. These are major tools for getting your layouts, essentially all your graphical reports ready. And then this tool, the table editor, is a major tool for okay, so the middle, well, what I should say is down below these major task bars is the ribbon essentially that exists in FlowJo. And you can see when I click on these tabs, right, the ribbon underneath kind of changes. And I'm not going to explain all of the different bands to you guys today, but the general idea is that if you're looking for certain functions that perform in different areas of the program, they're generally subdivided here by the different types of tabs. Okay, I'm going to show you where all the major functions are. Again, try to get your work done as fast as possible so that you do the least amount of clicking. And we'll, as I said, I'll talk about those kind of as we do the analysis. Also here in the middle pane, this section is all about grouping your files. So this is known as the groups pane. Groups are just like subfolders that exist in your workspace. So they help you sequester files that you want to do specific analysis on. And a lot of times when we run experiments, we have single stain controls, for example. In my case, I've got single stain controls, I have FMO controls, and then I have experimental files. When it comes to creating graphical reports, a lot of times I don't want my compensation controls ending up in the reports with my experimental files, right? I just want to show you know, in my experiment that the cells were responding to a particular stimulus or something happened to a particular subset of cells as a result of some condition applied. Okay, whereas the single stain controls, they're just there to generate essentially a comp matrix for us. Okay, and then the FMOs are used as gating controls. So we'll create groups for these guys in a moment, but the groups will result one is labeled compensation. So by default, FlowJo is going to have an all sample called immunophenotyping experiment, and it had a subfolder within it called compensation files. So when you drag in a folder that has subfolders, they will automatically be populated here as groups. Okay. All right. So when you bring your files in, the first thing you'll notice are these little icons that sit to the left of your FCS files. Okay. These little icons are telling you something or other about what's going on with the data set. Okay. The first one you can see here, right? Some of my files have this open square. Simply means that your files have a compensation matrix associated with them. Okay? If they have an open square, it means that there's no compensation matrix associated with them. In this particular experiment, we ran compensation on the machine, and then when we exported those FCS files, those exported FCS files pick up or retain the compensation matrix associated with them. Okay? You can generate a comp matrix in FlowJo itself by using your single stain controls and then apply it to the samples that you want. And we'll do that a little bit later in the webinar. Okay, if you want to inspect the comp matrix that's associated with your FCS files, you can always just double click on the gray grid, right? Or whatever color that grid happens to be. That'll open up what's known as the matrix editor. And we can kind of zoom in a little bit here in the bottom. Right, but in the top, we have a table here that shows you all of the spillover values for each one of the parameters into the various detectors. But more importantly, I think, is the chart or the n by n plot that you see down below. Okay, so here in this n by n plot, you're actually looking at two different sets of data, essentially. Okay, the blue dots represent your data uncompensated. So if there was no compensation associated with your files, okay, in this case, we're looking at the FMO for one of my samples here, right? The blue dots represent what that FMO looks like without any compensation being applied. If you actually untoggle this box on the right where it says overlay uncompensated, right, we just untick that. The black dots here, right, it removes the blue dots, and the black dots that are underneath represent the data as they look now with the current compensation matrix. So this gray grid corresponds to the gray acquisition defined matrix that's listed in the left-hand pane. Okay, so these dots, these black dots, right, are your data as they the middle portion of the positive population, right, roughly matches the middle portion of the negative population. Okay? On occasion, you'll see events that are, sort of crashing in towards the y-axis or maybe down towards the x-axis, right? Other times you might see a bend right in towards the center of the graph, okay, either along the x or along the y-axis, and it's a fairly obvious bend or a very interesting sort of relationship. So, for example, in this corner here, where we have PE Texas Red versus PE Sci 5, right? You kind of see this bending inwards of the blue dots. Okay, in this graph over here, where we have PE Texas Red versus PE. Right, you get something else, you get this little sort of 45-degree angle going. Okay, a lot of times when you see these streaks, it's another good indication, right, that compensation either has not been done or has been done very poorly. Said, if you're coming in here to inspect your matrix before you do your analysis, just make sure again that the positive populations here, either along the X or along the Y, are roughly matched up to, right, then the one in the negative here, in the middle of the negative. Okay, if they're not, you want to take your single stain controls and generate a new matrix to try and achieve that. Okay, and again, I'll talk about that a little bit later. One is your sample quality check. So the way to sort of invoke this or to do a sample quality check on your samples is to head up to the Tools tab here And in the first band, you'll see it labeled cytometry. There's this button that says check sample quality. If you click this button, what will happen is FlowJo will go through each one of your files, right? And it'll place a colored dot inside that little circle, okay? The colored dot means something about the acquisition, like whether it was smooth or not. So if you get a blue dot or a green dot, it actually means that the sample quality was pretty good. There's no major drift in signal, okay? But if you have a purple dot or a red dot, it means that there is a spike in the data someplace, and that could be caused by several different things, right? So you could get a spike in the data because of a clog, right? A lot of times if we don't filter our samples before we add them on the cytometer, right? A lot of times bubbles will actually trigger the purple or the red dot. And then the other option, or the other thing that sometimes happens is sometimes I've seen people change voltages during acquisition, or perhaps a laser is not is starting to peter out, and so either the voltage goes up or goes down, and you kind of see this, increased power and then weakening trend therein. And in that case, that will also trigger the sample quality check to flag the sample. Okay, so if you actually double click on the little circle icon in the bottom right hand, so this window will pop up, and in the bottom right hand section of that window, you'll see a bunch of graphs. And these graphs essentially correspond to each one of the parameters. The x-axis here is actually time. So we're just looking at the collection of this particular parameter, in this case, forward scatter, right, over time. And the little sort of double dashed lines, let's say above and below the green line there, those are the markers for where two standard deviations are either above or below that line. So if you get a spike that exceeds that double dashed line, you usually are going to get a red dot here. Okay, if it's touching that line or just below it, a lot of times you'll get a purple dot. Okay, so you can come along here, just inspect the parameters. If there's any, again, if you get a purple dot or a red dot, I would just go ahead and open this up and then just look to see which parameter will give you the best visualization of the drift in signal. So in my case, the most erratic channel here is probably side scatter, right? Maybe that or PE Psi 5A. And everything else looks pretty nice and smooth. Okay, and this one is a little bit more erratic than the others, but that's not a problem, again, because this has got a green dot, but we'll inspect this in a second. Okay, the other thing that's important about this pane is on the left-hand side, it contains a whole bunch of information about what is included in your FCS file. So the FCS file not only contains, the data about your cells, but it also contains information about, which cytometer was used. Okay, in this particular experiment, an LSR2 was used. We can see who the operator was. Okay, in this case, it was my colleague Tim Crawford. It was even done at the Institute or the University of Hawaii. Okay, so there's a bunch of metadata, or what we call keywords, that are associated with your FCS files, and then you can see what those keywords are and what the values of those keywords are in this left-hand pane. And we can actually use this information to sequester our files into different groups. Right? It'll help annotate the data so that we know, what went on during the experiment. All right, let's assume for a second that this was the problem. We had a problematic file. This, was a purple dot or a red dot, something like that. Just go ahead and double click on the file itself, right? And it will open up a graph window. Okay, in the graph window, what you want to do here, okay, is convert this axis here just by clicking on the little button, right? It's got this little arrow pointing down. Just click on that. If you had a gap, if you had a clog, right, you would see a gap generally towards the left end of the graph. There'd be some weird events on either side of the gap, and we would try to cut them out. Okay, if you have bubbles, a lot of times what you'll see is the trend of this data sort of curve upwards, and you'd have a lot of spurious events on the right-hand side of the graph. Okay, this acquisition is pretty smooth. We've got a little bit of thinning here in the middle. You can kind of see how it's red, 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 and then it kind of goes, you know, you don't have so much density of redness here, and then it kind of increases towards the end. Right, what you can do here is you can create a gate on the anomalous region. So this is not bad, but I'm just going to say for the sake of argument, right, that this section of the acquisition is anomalous and we want to chop it out. Okay, so we're going to go ahead and create a time gate using this rectangle tool at the top. So all the gating tools are located in the upper left-hand side of your graph window. You just click on the tool that you want to activate it, and then you just simply drag your mouse. You click and drag your mouse across the interface, right? And we can call this our time gate or something like that. All right, and just move that gate so that it's over, you know, spanning the anomalous region. If you want to tighten this up, you can move any other corners, right? We can move this up so that it goes a little bit beyond the top of this screen there and then all the way down to the bottom. Okay, now by default, FlowJo's gates are inclusive, which means that if I double click on this population, right, it'll show me what's in that gate in the next graph. Okay, so really all I did here, right, is I started on this, created that gate, then I double clicked in the gate and it shows me the population that exists in that gate. Okay, that's pretty standard behavior. And if you want to change this so that you chop out the data that are contained here, right, and completely ignore what's going on, and there are several options down here at the bottom of your graph window. Okay, the one that I'm going to look at here is called the and so that the downstream population exceeds, or doesn't exceed, it gets rid of whatever is in that gate. Okay? So that's how to create a time gate if you want to make it exclusive. Okay? All right. A little bit more about time gates, but for the time being, we'll leave that alone. Okay, now this little blue diamond here, this blue diamond is just an indicator of which graph window is open, right, or which sample you're looking at last. So you'll see the blue diamond sort of jump around as I create gates in the tutorial today. Okay, so once we've done the sample quality check, if we had to create a time gate on any of our samples, I'd go ahead and add those time gates, you know, make sure they're set up. Okay, and then the next thing that I do, the next step, is to go ahead and group the samples so that we don't get our, for example, our single stain controls mixed up with our experimental files or anything else. Okay, so the first thing that I'm going to do actually here is I'm going to go ahead and create a group just for the FMOs, because I'm going to create gates on the FMOs, and then I want to apply those gates to the experimental files. Okay, so FMO stands for fluorescence -1. This is a type of gating control that is used especially for a lot of terminal markers that we use in flow cytometry. Right? We want to know where the positive and negative. Okay, so we come up here to the top where it says create group. Go ahead and click that button. I'm going to call this my FMO group. I'm going to change the color here to something a little more appealing. Right, and then from here, if you just want that group, right, you can just go ahead and say create the group. It'll go ahead and place that group name here. It won't have any files in it just yet. Okay, but you can jump back to the all samples group, right, and then hold down the shift key if you want to highlight the samples you want to bring in. And then you just simply drag and drop that into the FMO group. Okay, and now they have those 7 FMO files that we have. Okay, the other thing that I want to do here is I would like to sequester all of my experimental files as well. Okay, in this case, I'm also going to create a group. Again, clicking on that plus at the top. Right, we'll call this experiment. I change this over to, again, some other color that's a little more appealing. Okay, but in this case, I'm going to make use of this sample inclusion criteria box. Okay, remember those keywords I was talking about, right, when we clicked on this little circle badge? I'm actually going to make use of the fact that the file name keyword, right, is different between all of the samples. So I have cell comps written here, right, FMOs written here, and then a whole bunch of LD something or other written for these files. So I'm actually just going to exploit the naming difference here to draw the right files that I want into the group. So I click on the little button here, tell it go ahead and find that file name keyword. Right, and that will create a group that contains all the files that have that LD in front of them. Okay, in this case, it has all my FMOs, or sorry, all my experimental files. These are local donors. Okay, I could do the same thing for FMO if I wanted to. It would probably work on those guys. Okay, if you had another keyword that you wanted to use, to label the samples, or perhaps you have a time point or a drug concentration or something like that you want to group the samples by, you could certainly use that keyword. Okay, I should mention here that and then you just write, it doesn't matter which one you choose. And then you could say add keyword, right? And this will present you with a new column. So I could just call this my new keyword for the time being. Right, that'll present you with a new column here, and you can essentially type in whatever value you The way to do that is go to the Configure tab. You'll see this Edit Columns button. If you click on this, it'll present you with essentially that list that we saw in the Inspect menu here. Right? It'll allow you to bring in those keywords so that you can, make adjustments, or you can visualize them directly in the workspace. Okay, so all of these files have a well ID, for example, associated with them. There are some other annotations that we added on the machine that I might want to look at. So, for example, the condition, or maybe the HIV status. Right? Maybe the stem condition, something like that. So I select the ones that I want by using the Control key or the Command key, and you just say Add Column, click OK, and then it'll bring all that information here. Okay? If there was any problem, maybe they were mislabeled somehow, you can always come in here and retype it. All right? If you right, you're descending. If you don't like the name that FlowJo uses here, you want to use something else as the name for the name column. The way to do that is head up to the preferences. So this little heart icon here is your preferences for FlowJo. Click on that. Right, and then you'll see this button where it says Workspace. You click on the Workspace button, and in the very top here, it'll have a box that says Sample Display Naming. Okay, from here, you can change the keyword that you use. Okay, so this keyword has to already exist, or you have to have created it in FlowJo before you can, use it for the name column here. But for example, I could say, I want to swap the name column here out for the well ID, right? So I'd come down, I'd click on the drop-down menu, and I would select the well ID, right? And then click OK, and then that will swap out what FlowJo uses by default with the well ID instead. Okay, and you can kind of see here that the samples are in a slightly different order. All right, click on this again. All right, to send it back to the original, I can say FCS default, which is the sample name keyword. Click OK, right, and it'll bring it back the way it was. All right, so we're the FMO group. We're going to start here, and I'm going to double click on that file. Okay, I'm going to return this back to what it would look like by default. So usually this is going to be forward scatter by working with cells that have a high refractive index, right, they're going to be higher on the side scatter than others. Okay, in the case of our lymphocytes here, lymphocytes are generally pretty low on the side scatter, and they're fairly small compared to, you know, say monocytes or some of the, well, granulocytes, I guess, are roughly the same size. Okay, but they're definitely smaller than a lot of other cell types that you may encounter. If you want to change the type of graph that you're looking at here, okay, maybe you don't like the pseudo-color. Pseudo-color is cool, I think, because it gives you density information, right? So the warmer colors are telling you where more cells are falling. The cooler colors are telling you that there's fewer cells in that vicinity. But you can visualize this differently by changing this over, for example, to a contour plot. The nice thing about contour plots is that if you have events that are pressed up against an axis, right, it's very easy to see them because the contour lines sort of bubble off of the axis. So it's a very, very nice little trick. We have a whole bunch of other types of plots, you know, if you want to. So by far the most popular types of graphs. And so that's what we're going to stick with for the most part today. Okay, you can smooth this if you want to make it look somewhat like a contour plot, and then you tick that show outliers, right, and it'll show you the lowest percentage outside the last rung. Okay, again, I'm going to return this to the dots, and then we'll begin our analysis. So I'm going to start my analysis here with the polygon tool here. Singlets are usually going to fall along this 45-degree angle. Right, and the doublets are actually going to be the clouds of cells that are sort of falling off of that 45. So I'll go here and I'll call this my singlets. Okay, and a lot of times to detect singlets, we use either the area and height, right? Maybe you use the height and width parameters. On occasion, you'll see -W. In my case, I don't have that available. I only have the area and height parameters. Okay, and you can do this again on the side scatter if you happen to have side scatter area and side scatter height or side scatter width. Okay, in my case, I don't have the side scatter height or width parameters, so I'm sort of limited to one singlets gate. But the idea is, you know, clean up the data as much as you possibly can before you begin doing your analysis, because we don't want to in there either. So we're going to take the next step here and chop out the dead cells. Okay, I double-clicked. I just double-clicked inside this gate to get to this population. And now from the drop-down here, I'm going to go ahead and change the x-axis to AARD, which is the amine reactive dye. It's very much like trypan blue, right? The live cells are the ones that exclude the dye, and the dead cells, right, are the ones that include the dye. So I'm going to go ahead and create a rather generous gate here. Right? Now I'm going to call this my live gate. Okay, I'm extending this gate a little bit, you know, out to the right because I can see a cluster of cells here. These probably are my monocytes because the monocytes tend to coat themselves with AARD, and we can test this a little bit later on. But AARD is the dye we're using, and monocytes tend to put that around them. They're not dead or dying, but they're very sticky. Okay, so I'm going to include them for this portion of the analysis, or at least have them in that gate. Okay, and then we're going to pop back here to our forward. All right, create something like this around this guy. You can be, you know, my general advice is be a little bit more generous in the beginning, and if you need to, you know, you can go ahead and tighten up these gates after you sort of witness what falls in them. Okay, after you create the lymphocytes gate, again, your analysis is going to be completely different than mine, or, you know, it might be similar in some aspects, but it'll diverge at some point. Okay, I'm going to take a look at the lymphocytes here, and then we're going to move on. In my case, I'm going to move on to CD3s. And we're going to see whether or not they're responding to a particular stimulus we've applied. Okay, a lot of times you'll see your fluorescent markers here, your fluorescent data sort of produce graphs that have populations that appear squished. Okay, you can expand these populations or rescale the data by clicking on the T button here and then say Customize Axis. right? And this will present you with a histogram that allows you to change the scaling, right? So I'm going to keep this graph off towards the right just as a, so we have it in immediate memory. Okay, I'm going to get rid of some of the positive space here since, we can make use of this real estate. And then to control the area around 0, right? This section between negative 10 to the third and positive 10 to the third is a linear portion of the scale. So a lot of your flow cytometry data, especially the fluorescent parameters, are going to be placed along what is known as a bi-exponential scale. So from 10 to the third to 10 to the fifth and beyond, you see a bunch of uneven tick marks. This is telling you that this section of the scale is logarithmic, right? This section here between minus 10 to the third and positive 10 to the third is linear. The whole purpose of this is to be able to see negative populations that are nice and round, right, with positive populations that are also nice and round. In the old days, we used to have to do straight log or straight linear and then kind of amalgamate it. But with the bi-x, it makes it easy. Okay, so we're going to chop off some of this stuff, and then we're going to blow up that area around 0 a little bit. Right, if you need some extra buffer space, you start to see some cells piling up on that far or on the y-axis. Right, just use a slider there. Right, maybe not so much. And give yourself some space. Okay, then click the apply button here. I'm going to apply it actually to both CD3 and the DR parameters, so both the X and the Y here. Click apply. You see how that blows up, right, the population. This graph looks very different than the one we had seen just a few seconds ago, okay, but the data are exactly the same. We're just visualizing it differently, and we're making it easier for us to create gates. Okay, so I'm going to go ahead and use the quad gate here, the curly quads. I like these curly quads. They have these curved arms that help you identify single positives, away from double positives. A lot of times you'll see this sort of trumpet shape or sort of this funnel-shaped population of cells, especially, as your data become brighter, it'll be either on the y-axis or along the x. And the whole purpose of these quads, right, these curves, is to sort of follow that funnel shape. And the funnel shape occurs because of spillover spreading. This is a problem that is intrinsic to the machine, and it happens independent of compensation. So compensation actually helps to reveal the spillover spreading, but it doesn't eliminate it, if you will. Okay, so we'll go after the CD3 positives. So all I did there was double click on the quadrant that I'm interested in. Okay, so I actually made use there of the little up arrow. The up arrow will take you to the parental population, right? If you keep clicking up, it'll take you further and further up the hierarchy. And as you notice here on the workspace, right, we have a whole series of gates that we've created. The hierarchy tells you what the dependency of happens in the lymphocytes gate, and of course all of the quadrants that are down below. Okay, if I double click on this, we'll move right on here to, well, let's go to CD4 versus CD8. Okay, this is an FMO, so we don't have really anything present here, but this allows us to create a gate. There's a little bit of carryover. You can see that dust kind of in the middle of the graph, right? But generally speaking, the FMO, right, allows you to create that gate to see where the boundary is between, the negative population and the positive. So the FMO tells you what the spillover contribution is from all the other dyes except the dye that you're working with. Okay. Now the CD8 gate, I'll go ahead and create the CD8 gate here real quick, and I'm going to show you a quick little tool that I hope you guys will make use of. Okay, I have all these gates. Okay, what you could do is you could drag and drop these gates onto the group, and that will copy the gates that you have to all of the other samples. Okay, but there's another tool here that I think you guys should make use of. If you just right-click on the gating tree, right, there's an option down here that says copy analysis to group. I guarantee you that if you use this function, right, it'll save you a ton of time. I don't have the time today to kind of discuss all of the great things about it, but take my word for it, copy analysis to group, and we'll actually be using this today in a few more examples here, okay? This is the keyboard shortcut that I use more than anything else in the program. Okay, so we go ahead and here say copy analysis to group. It takes everything. This one is an FMO for interferon, and I want to open up the CD8 positive population, right, that doesn't have interferon. So we'll click here and say, change the parameter over to interferon, and this will allow me to create a gate on that population. Okay, here's interferon. All right, we'll leave that alone. I'm going to move that just a little bit off towards the right. Okay, I have this new gate. It's kind of, you notice how that the color of the gate as I added it, right? It's in kind of black plain type font and everything else is in red, right? When we copied those gates to the group, right, those gates change color. They take on the color of the group. So this is telling you, when you see the red gates, it's telling you that each sample, right, has the gate in the same position. Okay, and there's no new gate. This sample, right, which is the only one that contains an interferon gate and it's in black type font, right, it's just telling you that this gate is new, hasn't been copied to the group yet. Okay, so guess what? I'm going to right click on that population and I'm going to say, use that function again where it says copy analysis to group, right, and it'll put it in the appropriate location here in the hierarchy, right underneath that CD8 positive gate. All right, the next readout population that we're going to work with here is perforin. So again, I'm going to open up the CD8 positive population, and then I'm just going to say, show me the perforin. And I will repeat the process, right? Create a gate on the perforin positive sample, and then come back here, right click on that and say copy analysis to group. Okay, and then the last one that we're going to do here is with phospho-ERK. So ERK is a MAP kinase. We are going to look at the phosphorylated signature on that MAP kinase. And I am going to label this as pERK. Okay. And again, I just right click on this, say copy analysis to group. Right, and there we go. All right. So I know this is an intro to FlowJo session. I could just layout editor, but there is something I would like to show you here as well. Okay, so we have CD8 cells that we found, and we've described a gate that defines them as being interferon positive, right, or phospho-ERK positive, or perforin positive. Okay, but it's possible to have a CD8 cell that is interferon positive and phospho-ERK positive and perforin positive. Okay, if you want to know what the triple positives look like, or how many there are, right, you use a function known as Boolean gates or Boolean functions in order to arrive at that. So what you need to do, if you want to know what the triple positives are, or the triple negatives, or any permutation thereof, right, you might be looking for cells that are interferon positive, phospho-ERK positive, but perf negative. What you want to do is go up to the Tools tab after you've highlighted the samples, and then go to the Boolean band. So gate and the AND gate, the symbol here kind of tells you what they do. Okay, the AND gate is an intersection between the gates. The OR gate is going to be a fusion between all the gates. Okay, it'll present you with a little window here that asks you if you want to rename the files. Just say OK, right, and it'll go ahead and produce the various the various combinations. So we have here, right, interferon positive, phospho-ERK positive, perforin positive. These are the triple positive cells, right? We apparently have 7 cells in this gate, which represent 0.017%, right, of the entire CD8 population. So that's what the statistic here is. Okay, the statistic is always the frequency of the parent. So triple positives are 0.017% of the CD8s, of which we only have 7 cells. If you want to look at this, double click on the gate, right? You can kind of see the little dots here. Sometimes if you don't have too many events, it's a little bit easier to see, again, using that contour. Okay, the contour plot will reveal the lines and where those populations lie. If you tick show outliers, if any other outlying cells are there, it'll pick it up as well. Okay, you can copy these gates just like you would anything else, right? Right click and say copy analysis to group if you want to keep it. Okay, in my case, we're not particularly interested in the triple positives or any combination thereof, so I'm going to leave it alone. Okay, we've copied our gates to our, our FMO group. It's a good idea after you do a copy to a group to go ahead and save your work. So click on a File tab, then say Save. Right? By default, FlowJo will create a time-stamped folder, and it'll also create a time-stamped workspace. Okay, so in my case, I'm going to go ahead and send this. I want to put it on the desktop just so it's easy to find. Okay, and then we'll say save here, and we'll go ahead and save that workspace. Okay, I can't believe it's already the 1st of March. Man, time is flying. Okay, anyway, we have these gates set up. Another thing that we might want to do before we continue the analysis here is add some statistics. Okay, so a lot of times with these terminal populations in particular, we want to tell people about, whether or not the cells are responding to stimulus or not. Okay, and a lot of times what we'll do is we'll add an MFI. Okay, MFI stands for median fluorescent intensity. Okay, it could also be the geometric mean, but the key here is when you add the statistic for the MFI, you just select the population, then right click and say add statistic, and it'll bring up this menu. And in the menu, you'll see that median is at the top. Okay, you also have mean. You want to use the median simply because the median resists the influence of outliers. And a lot of times in flow cytometry, when we're looking at data, we're looking at data that are plotted along a logarithmic scale, right? It may be bi-exponential, but here, in this case, from 10 to the 3rd to 10 to the 5th, right? We're on a logarithmic scale. So a single outlier that's on the far side here, right, is if you had chosen the mean statistic, it would skew the mean statistic very, very strongly towards the right. Same thing if you have a single event in the negative side, you'll see that the mean would be skewed very strongly towards the left. Okay, but the median is a very good indicator of where the central tendency, where this ball of cells happens to lie. Okay, so we're going to go ahead and select median. The other one that works as well is the geometric mean. So both of these guys are resistant to the influence of outliers. They're both very good indicators of the central tendency. Because they are calculated differently, they'll give you different results, but the general idea is pick one and stick to it. I like the median. We're going to take median interferon is the population. Since we had started with that, that's what ends up here. And then you just highlight the parameter that you want it associated with. Okay, so in this case, it's PE size 7, the interferon parameter, right? And when you click add, it'll add that little stat node to the workspace. Okay, and then we're going to change the population. We'll just get an MFI for our phospho-ERK population. Okay, so I change the population here, and I just say, all right, I want it for Alexa 488 phospho-ERK. Right, change the population again. In this case, we'll do it for perforin as well, and then change the parameter, click add. Okay, if you want to count or something like that, right, maybe for our CD4s and our CD8s, just go ahead and add the CD4 population, select count, right, and change it again. I'll add this to the CD8. The count statistic and the frequency of, these are independent of the parameter, so you can ignore this pane when you're adding counts or frequency of. Okay, but for all other stats, you're going to have to tell it which parameter you want it for. Okay, so I come back, I'm going to close this, I'm going to come back to the workspace, and I highlighted all the stat nodes, right, that I want. And again, I'm going to right click here and I'm going to say copy analysis to group. Okay, copy that over. Right. It goes to the appropriate location, gets copied to the group here. They have all the stat nodes, right, and now everything has it. Okay, since this is intro to FlowJo, I should mention, right, that if you come into a sample and you move a gate around, in this case, this is the interferon gate. If I come into this sample and I nudge the gate, right, this happens to be the no ERK sample. So let's actually jump down in the workspace to the one that has no ERK, and it should be indicated by that kind of blue diamond, right? The solid blue diamond is telling me that that's the one I'm looking at. Right, here's that interferon gate. You see how it's changed color now, right? Instead of red, it has changed to black. Okay, that means that this gate, right, the interferon gate here is changed in position relative to its neighbors. Okay, so if you want it to go back the way it was before, right, highlight it here in the workspace and then click the delete or backspace button. Okay, so if you say delete, it'll say, are you sure you want to delete this? It's kind of erroneous. You just say yes. Okay, because the population has been copied to the group, you can no longer delete it from down here. Okay, but that will snap that gate, right? It'll snap it right back to where it's supposed to be, where all its neighbors are. Okay, so I'm doing that again just so you can see that snap to. Okay, if you like the new gate's position, if that's where you want it to be, okay, again, highlight this gate and then use our familiar tool here where we said copy analysis to group. Okay, and that will update all the gates in all your samples. Okay, so if you click on this graph window, you'll see the little green arrows, right, on either side. Makes you go forward or reverse, right? And if I want to make this kind of go back a little bit more to how we had it in the beginning, I move that gate, go to the sample that has it, right? And then I'm going to say copy analysis to group. Okay, I want that to be the gate. Say yes, and I'll go ahead and replace it. Now, technically, I should be on this sample, the one that doesn't contain the doesn't contain interferon, to set the gate. So I'm actually going to do that there. All right. Now that we have these gates and our stat nodes, we want to copy this information over to our experimental files and finally start getting, to the point where we can begin deducing something about what's going on in our experiment. So let's go ahead here and collapse this gate, right? Collapse the very top gate. This will be the way to copy the information, the easiest way to copy information from one group to the next. Okay, so I'm just toggling that little black arrow, collapse it, and then you just drag that to the group of choice. In this case, we want it to go to our experiment group. Right, everything will come in collapsed here. If you want, you can click on the Edit tab and you can expand these so you can see, right? You can expand or collapse those gates right away. You don't have to go through each one individually. All right, so I'll leave this expanded. And then the next thing I'm going to do here is I'm going to go to the Configure tab, and I like to hide my stat nodes until I'm ready to work with them. So I'm going to go to the Layout Editor next to create my graphical reports, but I don't want to use my stat nodes in the Layout Editor, and I don't want to see them until I'm ready to go to the Table Editor. So I'm going to hide them for the time being. So if you go to the Configure tab here, you can say Hide Stats, and it'll make your workspace a little bit more, I don't know, a little bit more workable. And then you click on the L button here. Okay, so what I did there, let me close this again and show you. Okay, click on the L button here. This opens up the Layout Editor. Right, this is basically like a sheet, right? A sheet of paper that you can put all of your graphs on. Okay, so there's a million different things that you can do in the Layout Editor. I'm going to show you again some of the shortcuts to make your analysis as quick as possible so that you don't spend a lot of time, clicking on things and trying to find stuff. And if I'm interested in showing in lab meeting, something about my cells, especially the interferon positive population here and how they respond or not to stimulation, I just drag that terminal population in, and then I'm going to make use of a nice little shortcut here. So if you right-click on the plot, okay, once you bring it into the Layout Editor, go ahead and right-click on it and say, here's this option for Ancestry. Okay, the Ancestry will show you a nice panel here that contains all of the gates, right, that led to the population that you're looking at. So these are all of our gates, right, that we've used. Right, here's the singlets, right, live, lymphocytes, blah, 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 all the way on down. Okay, the other thing you can do here is you copy this, move that off to the side. I'm going to delete this just so I can show you. You can also show the backgating here, so you click on the backgating. Maybe if you want big dots, you can say show large dots, right? And you have the backgating information included. Okay, backgating, at least in this sense, right? Backgating is just taking this terminal gate here that we're using to define the cells as being interferon and CD8 positive, right? But for simplicity's sake, let's just think about it in terms of interferon positivity. Right? We're just taking that terminal gate and we're projecting it backwards through our analysis. Right? So if we walk back one step, we see a whole bunch of red dots in the parental CD8 gate, which is what we would expect. But we also see some red dots that are falling outside of that gate. Okay? These red dots, these are cells that would be considered, right, interferon positive by the gate that we have down here. But they're not being quantified down here because, right, the parental gate here is sort of limiting. Right? We'd have to come back to this population and extend that gate to the right if we want to quantify them down here. Okay, that's essentially what the backgating is telling you. It's very useful for asking, the question, where else in my hierarchy can I find cells that are positive for a given antigen? Okay, and in this case, again, we're talking about interferon positivity. Right, we see here we went after the CD3 positive population in our analysis, but you can clearly see that the CD3 negative population here, which includes B cells, right, NK cells, maybe monocytes, things of that nature, right, are also positive for interferon. So this is just telling me that using that definition, there's a whole bunch of cells that I'm not capturing, right? There are other interferon expressing cells elsewhere in my analysis. Okay. Walk it back to some of the more quality control gates, you can kind of see that I've captured the vast majority of my lymphocytes are in that, sort of central region, and they, a lot of them there, they sort of spread the gambit, I guess you would say, of the forward scatter, right? So we have a decent gate placed on those guys. Right, we're picking up maybe some monocytes or maybe some cells here that might be heading towards, heading towards death. So if I wanted to, I might tighten this gate or maybe change the gate so that it, kind of makes an angle there so that it picks up the monocytes that are likely down here and maybe excludes some of these guys that are not part of the monocytic population. Okay. The other thing here too is we can see we don't have too many doublets that are interferon positive, which is good. All right, so once you have that, okay, we have a basic, we have a basic setup here. You can use the page delineators, right, to kind of put how many plots you want on a given page. You just go to where the mouse intersects, where the two lines, the dashed lines intersect one another, and you can see how my mouse changes from an arrow to a plus. And you move that, you move it far enough, you get either portrait or landscape, depending on how you want. Okay, and then from here, if this is the setup that you would like, you can go ahead and choose the type of export that you want. So by default, FlowJo sends it to itself. So it just says new layout here. Right? But you can also send this to the printer, which is the one that I like probably the best. When you do that and you click the Create Batch Report button, what this does is it presents you with the printer spool sort of menu, right? So I have my plots on a page here, and I can put them in different rows, right, and columns, right, however I want to arrange them. And then I could click the Print button here. And when I do that, I have an option to save this as a PDF if I want, okay? So it's a very versatile little tool. And of course, if you go ahead and print this, you get a nice little hard copy that you can put in your notebook. Okay, the PDF, when you export it from here, give you 300 DPI, which is generally what the journals are looking for. So it's pretty useful that way. Okay, if I go back to new layout here, and I'm just going to set the orientation to go in one column going down, so it's easier to see. And then I click this Create Batch Report, right? I could also use this button over here, the little cog. They do the same thing. So click on that button. And what this does is it produces a report, right? A batched report for all of your files. So this is sample number 1, the one that we started with, right? And then if we scroll down here, right, this is sample number 2. Right, sample number 3, sample number 4, 5, 6, blah, 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 all the way through. Okay, so just by clicking on that button, it produces a batch report. So you don't have to keep dragging in, graphs to see what they look like from sample to sample. Just click the batch button. It's just like copying your analysis to the group and everything gets it. You click one button here and it gives you a report for everything you have in the group that you're looking at. In our case, we're looking at the experiment group, and you'll notice, right, in the experiment group here, all of my samples, right, are the experimental samples. None of these are my FMOs, and none of them are my single stain controls. Okay, so I can send a report. I can give this to the boss and say, these are all, these are the results from the experimental samples, right? You can see here that in this particular patient, right, we definitely have some response to interferon, right, compared to this patient or this condition. Okay, so that's like a simple batch. Another thing you can do here, right? A lot of times we want to compare one population to the next, or one sample to the next, right? So I just click the little plus button here on the left-hand side. This will create a new sheet for you, and you can create a new report. Okay, from here, what I would like to do in my experiment, I've got a non-stimulated sample, a 20-minute, a 1-hour, and a 2-hour stim. And what I would like to do is just compare those stim conditions for each one of the patients that I have. So I have about 4 or 5 patients in this analysis. They have different numbering next to them, but there's about 4 or 5 of them we can investigate here. I've got 20 samples. You divide that by 4. That means I've got 5 patients. Okay, so what I want to do is I want to compare, let's say, I'm picking on the interferon positive population because I know this data, and the interferon positive population is the most interesting of the 3 that I've drawn gates for. So we're just going to stick with that for the time being. Okay, to overlay the populations, all you have to do, right, is either drag and drop the population into the layout editor and then drag it on top of the graph that you have, right? Or, if you straddle these guys pretty well, you just click on that population and then you can drop it directly on top of whatever you have, right? So this is essentially the setup I would like. Okay, I'm comparing the non-stim, the 20-minute, and the 1-hour and the 2-hour stims together in this graph, all for patient number 1. I'm going to move the legend in this way. If you want to change the axes, okay, instead of looking at this as a dot plot, what I'm going to do is I'm going to change this over to a histogram. So you just hover your mouse over the label, right? Not to the left, not to the right, but right on the label here. And then you right click. Okay, or on a Mac, you're going to control click there. And you can just change the axis to histogram. Okay, and it'll show you a histogram here. By default, they're going to be sort of smashed on top of one another. So what you want to do here is you want to change this, right, and say, right-click on the plot and say histograms, which will appear at the very bottom, and then choose the offset that you want. So I like, full offset's pretty good. The other one that I like is the stagger offset, which I'll show you in a second here. Okay, but immediately you can see from this graph, right, now that we've separated everything, I can see that the 1-hour and the 2-hour stim conditions, right, are responding to my my stimulation condition, whereas the non-treated sample and the 20-minute stim, nothing is going on. Okay, I'm going to change this to a stagger offset, just because I think it looks cool. Okay. And you can kind of move around that angle there to get an idea of, the landscape a little bit better. You can always rearrange these guys, right? If you want the non-stim to show up in the forefront, you can. You can change the color of it if you want, right? Make it gray or something like that. I don't know. It's entirely up to you. And you can change the line style and everything else. So pretty much if you just right-click on whatever it is that you're looking at, a lot of times you'll find the tool that allows you to change a lot of things about it, right? We could have it so that it's not tinted, right, or fully tinted, stuff like that. Okay, once that's set up, the next thing I have to teach you here is just to tell FlowJo how to create the batched report. Okay, so we have the samples set up. You'll notice here that everything is kind of grayed out. Right? You got to grayed out. The batch button is grayed out here, and it's grayed out on top. Right? What we have to do here, if we want to get the batch report to work, we have to change the iteration options. Iterate just means how to order the samples. So FlowJo right now doesn't quite understand how you want to arrange the samples. Okay, so we're just going to change the iteration options here. We're going to change it from off to panel. So anytime you have 2 or more samples overlaid with one another, you're going to want to change the iteration options to panel, at least panel. Okay, panel or keyword panel is the easiest. Okay, when you do that, you'll see that your graph gets blown up, but don't worry. Okay, all we have to do is make a little adjustment here. So this little box opens up to the right when you choose that option. And this box, right, is asking you how many samples you have overlaid with one another. Okay, in our case, we have 4 samples overlaid, so I'm going to tick that box up to reflect 4, right? And then my graph returns to how it was when we first created it. Okay, now you'll notice that the batch buttons return, and again, right, what I can do here is I can make this batch the report and columns going down, right, and I'll have this. Essentially what's going to happen is it's going to show patient number 2's data, then patient number 4, right, patient number 12, blah, blah, blah. Okay, once you have the data here, it's up to you if you want to, you know, again, you can batch this, as I said, to the printer, which you can get a PDF. You can also get a hard copy. You can also batch this directly to a PDF. You can batch to PowerPoint if you want. You're going to get an individual slide for each one of the samples. The problem is, a lot of times you're going to have these objects, when you bring them into PowerPoint, it's going to treat them as an image, and you won't be able to ungroup them. So what I would do in that case, like if you need to ungroup the objects, what I would do is go ahead and create the batch report to FlowJo, right? So we just tell this to go to new layout, then we click the batch button, and when we get this report, okay, from here, what I would do is go ahead and click on the File tab, and then you'll see this option where it says Export Image. When you click on that button, right, there'll be several image options for you here. The one that I like is the SVG. SVG stands for Scalable Vector Graphic. It'll allow you to ungroup the objects, right, in Adobe Illustrator or Inkscape. There are several PC programs that will also open up SVG files and allow you to ungroup them. Okay, you can also export here, you know, you can also save it here as a PDF or a TIFF, right? And these TIFF and PDF are going to give you 300 dpi. The SVG, the other nice thing about SVG is, in terms of the resolution, is that you can expand or contract the graph without losing resolution. So it's a very versatile choice here. Okay. All right. So that's what I wanted to say about the layout editor. We're, you know, a little bit, we're just past 11 o'clock here, and I kind of want to rein this in so that it doesn't go too long. I know you guys have a lot of work to do today. So what we are going to do, right, is go from here. We're going to close this, right, and we're going to open up the table editor. Okay, so we had the graphical reports, right, in the layout editor. Now we're going to go to the table editor to create our statistical reports. And I'm going to make a quick check here just to see. It shows that we only have one person. Okay, well, that's good. So as long as we have somebody here, that's all that matters. All right. So the next thing that we're going to do here is go to the table editor. Okay, the table editor is where we're going to go to export our Excel file or our CSV file. Okay, so you click on the table editor, open this guy up. Right, it works very much like the layout editor. It's kind of like a sheet. You go ahead and return to the workspace here. I'm going to toggle my stats right on again. Okay, and then in this case, I'm just going to take those stat nodes and I'm going to drop them into the table editor. Okay, so you just drag and drop them here. You can bring in populations if you want, but the default statistic associated with this population is going to be the frequency apparent. Okay, so if you want other statistics, I recommend that you add them first here as a stat node and then drag them in. Okay, I am going to make these guys look a little bit slick, so I'm going to go ahead and highlight the stats that I want to heat map. Okay, in my case, I'm going to choose the MFI for the 3 markers, and then I click on the Visualize tab and then use this heat map function. Okay, and now if I just click this little cog button, right, or I go to table editor over here and I say create table, right, these buttons do the exact same thing, right? Click that button and it'll give you a table, right, with all this, the selected statistics heat mapped. Okay, so here's the MFI for interferon, MFI for phospho-ERK, and the MFI for perforin. And you can immediately see, right, just basically what we saw in the graphical reports. The first two stim conditions, non-stim 20-minute, nothing is going on, right? The warmer colors here indicating that something is happening, right? 1 hour and 2 hour, we definitely have some activation, okay? With phospho-ERK, right, you have non-stim, nothing going on, 20 minutes, something goes up, and then it begins to go down after 1 and 2 hours, which is kind of expected for a MAP kinase with a phosphorylated signature, right? Same thing, that pattern repeats for each one of the patients that we have, okay? Perforin, on the other hand, doesn't seem to really increase much as a result of stimulation. It's really the expression here is varied more or less by patient than it is by stim condition, okay? You can copy paste this directly to Excel if you want, just Control-C, Control-V. You don't have to highlight anything. You can head up to the little edit if you want and say copy table to clipboard, and then you just paste it into Excel. Right, that's one way to do it. The other thing that you can do is back here on this page, you can say, change this from display to file, right? And then change this to either a CSV or an Excel file. Make sure you know where it's going, right? A lot of times this will go to a place that maybe you don't want it to go to, right? And I can call this, I don't know, Jack's table today, right? And say save, and then you have to click that button where it says create table, and you'll hear the chime, and then that will show you, right? That will create that table for you, and in my case, it's called Jack's table today XLS, right? If I double click on this, it'll open up Excel, right? And I'll have all those MFIs and the percentages and so on and so forth that I had in the table editor. All right, so that's the table editor, pretty straightforward. You know, you can create multiple tables just by clicking on the little plus button there, right? So you can have a B cell table, a T cell table, monocytes, whatever it is that you want, and divvying them up any way you like, okay? All right, let's talk about compensation. So the compensation group, again, is automatically trying to draw files in based on the naming. So if you double click on the comp file or folder or the group, you'll see that it's looking for any file name that contains the text unstained, right? Or any file that contains the text compensation, or in this case, just comp. Okay, if your files aren't labeled comp or unstained, don't worry. Okay, you can always go to whatever group they're in and just simply drag and drop them into the comp group. Okay, in this case, I dragged a sample that I had in the experiment group into the comp group, and now it sits there. Okay, for the time being, I'm going to get rid of that one, okay, because it doesn't belong there, but that's just to show you that you could do it. All right, now that you have your single stain controls here, right, you have to run the comp wizard in order to generate a matrix. So go ahead and click on the rainbow M here at the top, right? And then this will show you a little progress bar in the bottom left. And when it finishes, it will show you the comp wizard interface. Okay, and the comp wizard interface here, basically what's happening is all your parameters are going to be listed on the left. You're going to have a colored dot on the left-hand side. The colored dot is just telling you whether or not the sample name, which is here, and the parameter name are the same or not. Okay, in the case of the yellow dot, I have a parameter name, pack blue. I have the file name PB, so it gives me a yellow dot. In the case of AARD, right, AARD appears in the name of the control file. It also matches the name of the parameter, so you get a green dot. It's just telling you that FlowJo has matched up these parameter names and file names. Buttons essentially are showing you which sample FlowJo is using for the negative. So in this case, it's actually using the unstained cells. Okay, so cell comps, unstained, US cells, blah, blah, blah. This is what it's using for the negative. Okay, and for the positive, it's actually using the AARD sample and a gate that it has created for that particular population, right? And so on and so forth. Okay. If for whatever reason FlowJo got it wrong, you can always click on the button and choose the gate, right, or the population, or sorry, the file that ought to be used. Okay. In the case of Alexa 647 here, I have a parameter that was left blank. We had a channel that was open that we didn't actually use. I'm going to go ahead and drop this from the parameter list, right? Because it doesn't contribute to compensation here. So we just say remove that parameter and it'll dump it. Okay, and now this information that appears in the negative and the positive column essentially translates to what you have down here in the lower pane. So on the left-hand side, the sample is written in blue, right? Cell comps, US, unstained, blah, blah, blah. Right? These two match one another. Okay, and this guy over here, cell comps AARD, is essentially telling you which sample is being used for the positive. Pardon me. Okay, so the positive and the negative, they're just essentially overlaid here in the middle, and that's the graph that's being shown here, okay, as a histogram. The negative population is indicated by a solid blue histogram, and the positive population is going to be shown as a green untinted histogram. Okay, so for your single stain controls, you should always see separation between positive and negative. In this case, the AARD parameter, the positive gate, is definitely, you know, overlapping with the negative, and so this particular parameter would not be compensated properly. So we have to make a change here. Right? In order to do that, just double click on the graph that you want to change. Okay, in my case, I'm looking for that positive population, right? I need to find some cells that are positive for AARD in order to compensate it properly. So we just double click on the positive population here. Okay, and then from here, click on the gate and then move the gate into a location where you think you would find positive cells. Okay, so I know that dead and dying cells are going to be off towards the left, right, and a little bit high on the side scatter. Okay, and the minute I move that gate, you can see suddenly the histogram changes here, and you can see those cells move off towards the right. And if you kind of want to explore, you can see essentially what happens here, right? I move higher, 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 right? And we're getting some very positive cells here. A lot of dead cells that are positive for AARD, right? And what happens if we move it over here? Okay, these guys, again, these guys are the monocytes, right? And they tend, again, to pick up that dye a little bit, though they're not necessarily dead. There are some dead ones over here that are really bright. But the vast majority of the monocytes, they're healthy, but you can see here how they have that positivity, right, associated with them. Yeah, and another set of cells on this side. These guys are not, they are not very positive for AARD. All right, if you go in the bottom corner here, this is where you're going to have a lot of debris as well. Okay, you see some positivity. I'm just going to go after, I can find, you know, quite a bit more. So I'm going to go ahead and move this gate in that location. Okay, I'll leave the gate where it's at. Maybe trim this ranged gate to make sure that I get the positive taper at the end there. All right, just extend that a little bit. And then you can see here the negative population, right? Again, it's on a nice healthy population of cells, which is fine in a sense, okay? But because we have this cell here on some dead and dying cells, we want to make sure that the autofluorescence on these cells essentially matches the autofluorescence on these cells. And I guarantee you that it's going to be different. Not going to be hugely different, but it's going to be different. Okay, so what I want to do is I essentially want this gate to be translated or transferred over here for the negative. Right, now I could come in here and kind of move this gate off to the side to try and match the position, but if I want the exact position, right, so that these guys are exactly the same, the way to do this is to go back to the workspace. Okay, so we're looking at the AARD parameter. Okay, we're using cells for the compensation of this particular parameter, so we're going to go to where the cell comps are, which is down here. Okay, here's cell comps AARD. This is the one that we're using for the positive. There's that size gate. Right? Now all I'm going to do is I'm going to take this gate and I'm going to apply it to my unstained cells. So I'm just dragging and dropping that gate onto the unstained population. Let's see. gives you a warning here telling you you're going to replace the gate that's already existing on that unstained population. And you just say yes. Okay, and then what happens is, all right, that gate gets placed on those cells. Okay, and then it's just a matter of going through the rest of your samples. You see how the negative here too, you see how it has this kind of double spike, right? The autofluorescence here is a little bit higher, right, for dead and dying cells. These are unstained cells, so there's no AARD in the medium, but you can kind of see how, right, their autofluorescence, right, is increased. And we just want to make sure that the autofluorescences of these guys are pretty decently matched, okay, between the negative and the positive. All right, so from here, right, we'll go on. There's another problem here, right? We've got a overlapping positive gate, and then the negative gate is the one that has been placed in that position. Okay, if we want to make this so that this gate appears over here, there's a little trick we can use. Again, this is another cellular cellular control. So I'm going to first fix this just by moving that ranged gate right over this, and then for the size gate, come back to this. And what I'm going to do in this case is I'm going to rename this gate. I'm just going to call it, I'm going to go, I'll show you where that is. Okay, so there's a shortcut that I always use, but it's just Control R. I'm going to say rename. And the reason I'm going to rename this is because they're all labeled size. So if I drag this to the unstained group, it's going to replace the gate that I had placed on the AARD parameter. So I'm just going to call this size 2, right? I could have called it, I don't know, size APCH7 or something like that. Okay, but by making it size 2, right, now I can just drag and drop that onto the unstained cells, right, and it'll have size and it'll have size 2, right? It won't replace it. And then I get the gate at least in the same position that I want, and then I just make sure here that in the Alexa 700 channel, right, I just want to make sure that that negative gate that we're using is the size 2 gate. Okay, right now it's using the one that's labeled size. You can kind of see that the blue gate here, there's a blue tinted gate, right, this guy, and this one is in black. So basically this one is the one that's being used here, but we want to use the guy down here. So we just click on that button, right, say size 2, right, and that'll swap that out for the negative population. Okay, and the positive pop, I'm not exactly sure why it just dropped my positive population, but we got to come back here. We'll go to this guy and we'll say size 2 positive. That should return it. Okay, there it goes. It needed a little refresh. Okay, and we just add that one right there. All right, now this one is using the size 2 gate. That's the problem, sorry. The size 2 gate here that it's using is actually on the stained sample. So what we want here is we want to use the unstained sample, and we want the unstained size 2. So you see how it doesn't appear here in the list, right? We have the Alexa 700 size 2 gate, and we have the unstained size gate, right? But we don't have the unstained size 2. So when that happens, my favorite move here is just to force whatever you have in the workspace on whatever FlowJo has. So I'm going to take this gate here underneath the unstained cells, right, and I'm going to drag it into the appropriate location. So I drag and drop that there, right, and that forces FlowJo to use the correct gate. Okay, now we're using that size 2 gate on the unstained cells, which is proper. And we're using the size 2 gate on the positive cells, and then the range gate has been fixed. Okay, so when in doubt, just drag and drop what you have into the stuff here, because sometimes that list just doesn't refresh fast enough for us. All right, once you have the matrix, right, this is done. You can go ahead and apply this matrix to your samples. So if you click on Apply to Group here, I can send this to my experiment group, right? When I click that button, the color of the matrix now that shows up next to those samples is going to be this kind of crimson red. So if I head back to the experiment group, you'll notice that that little gray grid has changed to red. If I go to the FMO group, you'll see that they still have the old gray matrix associated with them. Okay, so if I want to send this matrix to the FMO group, you click on this and just say FMO as well. Okay, and now the FMOs will also pick up that grid. All right, if you want to view your matrix, go ahead and click View Matrix. All right, you'll see your comp matrix highlighted here, that crimson red one. Here are all the spillover values. It's going to look very similar to what you had done previous, maybe on the machine, all right, with some changes. All right, but then once they're here, all right, if you want to compare the matrix that you made versus the one that you did on the machine, you can just simply highlight these guys by holding down the Control key or the Command key. Right, and it'll subtract one matrix from the next, and then you can kind of see where the differences are. Now, again, I think these matrices are pretty good. Right, we have some differences here when it comes to, it looks like PAC Blue and AARD, right, and PE size 7. There's a little bit going on here. The red dots are shifted relative to the gray dots a little bit, right? We have a little bit of a dip here on the negative population in PE well, actually, this is not for PE size 7 so much as it is the PAC Blue channel. So PAC Blue is kind of what we have along the y-axis, right? And it's dipping down a little bit. So the red dots are shifted down slightly as a result of the new matrix that we generated, right? And they're also shifted down when looking at the cells, if you were to look at the cells in the Alexa 488 versus PAC Blue channel. Right, but for the rest of it, right, there's a lot of decent bit of overlap between the red and the gray, which is kind of expected, but this is a nice way to look to see whether or not your comp matrix that you generated on the machine is exactly what it ought to be on your data set. Okay, so this is just a way to do sort of quality control on your matrix. All right, and then from there, if you have to, you can always click Edit here, say yes. We don't recommend that you do this, but it's in the copy. When you say Edit, it'll create a copy for you. And then here, you can actually change the values around, right? So in this case, I might make this a little bit high, right, for Alexa 700. And you see how when I add that value, make it a little bit higher, you see how it overcompensates here, right, along the Alexa 700 parameter. Okay, it does this for a few other channels too here, right? PE size 7 is affected, right, and maybe a few others. Okay, so once you have that, if you want to apply this new crazy matrix, right, the one we changed the values for, you have to click the M button and then drag it onto the samples of choice or the group, right? So in this case, I could add that to the FMO, right? If you want it to go back the way it was before, go back to the acquisition defined, click on the M button, and then drag and drop that onto the cells. So this M, right, you can drag onto either an individual file, right? In this case, LD1 will have, the red, and the first LD1 will have gray. This way you can have multiple comp matrices in the same workspace, and you can have samples that come from very different experiments all in the same workspace, and you can generate the comp matrices that you need. All right, the last thing I'll mention here before we exit out, always again wise to save your work, but the advanced tools are located here underneath the Workspace tab. You'll see this populations band, and there's the plugins. Okay, you have several plugins that come with FlowJo by default. On your system, you will likely see Cell Ontology, Downsample, FlowMeans, Spade, and Tisney. Okay, Tisney, Spade, and FlowMeans, these are algorithms that attempt to identify populations of cells based on a lot of common markers. So if you're working with more than 6 colors, I would say that any of these algorithms are pretty useful for identifying populations of cells in your data set without you sort of, instructing the algorithm on how to find them. It's an unbiased approach, okay? The algorithms are different in the way they calculate differences between cells, and the way they display the differences is different. So Tisney will just give you a 2-dimensional plot. It's an extremely powerful tool for witnessing changes in the system. So I really, really encourage you, if you haven't already, check out Tisney. It's a very powerful tool. Okay, Spade and flow means are also pretty cool. Spade gives you a nice little graphic that shows you a relationship between the populations that it finds. Flow means is fast, but it doesn't give you any relationship between the samples. And then the downsampler is just a gate that allows you to create a set of cells, a fixed set of cells. So for example, if you want all of your lymphocytes, instead of being 210,000 here, and in this sample there are only 197,000, you can reduce it by using the downsampler. You could say, okay, make everything have just 20,000 events, right? You could say, just make my lymphocytes gate have 20,000 cells in it. So you just activate the downsampler, you tell it how many events you want, and it creates a gate for that number of cells. I could call this my lymph down. And we'll go ahead and calculate, and then it creates that population here for you, which has only 20,000 events. Okay, and then cell ontology is a nice little tool that queries a database, the cell ontology database, to find out if your cell has been published or not. So for example, if you have this interferon positive cell, it'll say, okay, your marker is interferon positive, CD8 positive, maybe CD4 negative, and CD3 positive, so on and so forth. And it feeds that information to the database, and then the database will spit back some information telling you what the name of that cell is, if it exists in the database. Okay, with that, I will say that ends today's session. If you have any questions, by all means, either use the chat box or unmute your mic, and I'd be happy to answer any questions that you have. All right, well, thank you very much. It looks like there's nobody left. All right, let's stop this.